You have already learned the four postulates that characterize Mendelian inheritance, such as unit factors in pairs, dominance over recessiveness, segregation, and independent assortment. However, some inheritance patterns do not follow, or disobey one or two of these four postulates. Those inheritance patterns have been grouped together, and are referred to as non-Mendelian inheritance patterns. The different types of those inheritance patterns will be discussed in this video lesson. The specific topics include the following. Under epistasis, we have dominant and recessive types. Under allelic interaction, the topics are incomplete dominance, multiple alleles, codominance, and lethal allele. Lastly, under sex-linked traits, X-linked dominant, X-linked recessive, and Y-linked traits will be covered. Let us now compare Mendelian from non-Mendelian inheritance. The main difference between these two sets of inheritance patterns lies on the four postulates that characterize Mendelian inheritance. One or two of these postulates are not followed in one of these inheritance patterns. Let us start with dominant epistasis. Among the four postulates, independent assortment is not followed in this type of inheritance pattern. How is this so? We will find out in a short while. Dominant epistasis is characterized by having complete dominance at both gene pairs, but one gene, when dominant, epistatic to the other. Let us break this statement into chunks so that you may understand it better. The phrase gene pairs indicates that two genes, for example, gene A and gene B, are involved in this type of inheritance pattern, and there is complete dominance at both gene pairs. As shown in the illustration, even one copy of the dominant allele for gene A is enough for the trait to be expressed. The same thing is true for gene B. The presence of one copy of its dominant allele will be enough for the trait to be expressed. But, when one gene, let's say it's gene A, is dominant, it will be epistatic to the other gene. Epistasis means to stop. Hence, whenever the dominant allele for gene A is present, it will stop the expression of gene B, both dominant and recessive alleles. Gene B will only be expressed when the recessive form of gene A is present. The occurrence of dominant epistasis was observed in summer squash. Two genes are involved in determining the color of the squash. The dominant allele gene A codes for white color, while the recessive allele codes for the presence of color other than white. On the other hand, the dominant allele for gene B codes for yellow color, while the recessive allele codes for the green color. Take a look at the table, which shows the symbolic representations of the resulting genotypes when two parents, heterozygous at both gene pairs, were mated. The numbers found on the ratio column may seem familiar to you, right? These numbers indicate the phenotypic ratio in a typical dihybrid cross. However, due to the influence of gene A over the expression of gene B, we cannot expect the same phenotypic ratio for this given cross. Let us now apply the assumptions in dominant epistasis to predict the resulting phenotypes for this cross. Whenever the dominant allele for gene A is present, it will stop the expression of gene B. So, the resulting phenotype for this, and this, is white. In the case of the third genotype, since gene A is in its recessive form, gene B can now be expressed. The dominant trait, yellow, will be expressed. Since gene A is also in its recessive form, in the fourth genotype, and gene B is represented also by recessive alleles, the recessive trait, green color, will be expressed. The resulting phenotypic ratio for this cross is 12:3:1. Let us now proceed to recessive epistasis. In recessive epistasis, there is complete dominance at both gene pairs, but one gene, when homozygous recessive, is epistatic to the other. Again, two genes, A and B, are involved in the expression of a certain phenotype. When the dominant allele for gene A is present, the corresponding trait it codes for will be expressed. The same thing is true for gene B. However, when one gene, let's say gene A, is present in its homozygous recessive form, it will stop the expression of both dominant and recessive alleles of gene B. The occurrence of recessive epistasis was observed in mice color. The dominant allele of gene A codes for the presence of fur color, while its counterpart recessive allele, when expressed, will give rise to an albino. On the other hand, the dominant allele for gene B codes for agouti fur color, while the recessive allele codes for black fur color. When two parents which are heterozygous at both gene pairs were mated, the phenotypes of the resulting offsprings could be deduced, based their corresponding symbolic representations, shown in the table. Let us now apply the assumptions of recessive epistasis, to predict the fur color of mice, born of the two heterozygous parents. In this case, gene A is epistatic to gene B. For the first genotype, since gene A does not occur in its recessive form, there is no epistatic influence exerted by gene A on gene B. Dominant allele for gene A is present, which means the presence of color on mice fur is allowed. Which color then will be expressed, agouti or black? Since the dominant allele for gene B, which codes for agouti color, is present, the resulting mice with this genotype. For the second genotype, no epistasis will also take place, since the recessive form of gene A is absent. The presence of the dominant allele of gene A allows the expression of color on mice fur. Since gene B is present in its homozygous recessive form, which codes for black fur, the resulting mice with this genotype will have black fur. Let us take a look at the third genotype. Gene A is present in its homozygous recessive form. Therefore, it will stop the expression of the dominant allele for gene B. The resulting mice will then have albino color. The same thing is true for the last genotype. The modified ratio for this cross will be 9:4:3. 9 agouti mice, 4 albino mice, and 3 black mice. Now, let us explore the different types of inheritance patterns under allelic interactions. In allelic interaction, only one gene is involved. In incomplete dominance, which among the four postulates is not followed? The answer is, dominance versus recessiveness. Incomplete dominance is characterized by having heterozygotes, which are phenotypically intermediate, between two homozygotes. A classic example used to illustrate the occurrence of incomplete dominance is that of petal color among snapdragon flowers. Snapdragon flowers commonly occur as either red or white. If we want to represent these two traits using symbols, what symbols should we assign for each trait? You might readily answer, assigning capital letter R to refer to the red trait, while a lowercase letter r to represent the white trait. However, the dominance of red allele over white allele is incomplete. This suggests that it is not proper to represent white trait using lowercase letter. In this case we will also use capital letter R, but to distinguish it from red allele, we will place an apostrophe beside it, and it is read as R prime. This illustration shows the resulting phenotypes, corresponding to certain genotypes. An individual which is homozygous dominant, will have the dominant trait expressed as its phenotype. In the case of snapdragon, a homozygous dominant plant will have red flower. Meanwhile, a homozygous recessive individual will have its recessive trait to be expressed as its phenotype. Again, in the case of snapdragon, a homozygous recessive plant will have white flowers. However, a heterozygous individual will not bear the trait that the dominant allele codes for, as in Mendelian inheritance. Instead, resulting phenotype will be intermediate between red and white colors, which is pink. To further illustrate that point, let us see what will happen when homozygous red and homozygous white parents were crossed. All plants belonging to the first filial generation will have pink flowers. Now, what will be the resulting phenotypes when two plants producing pink flowers were crossed? Please pause this video for a while and try to identify the resulting genotypes, phenotypes, and phenotypic ratio from the given cross. Resume playing the video once you've finished answering. Now, check if you got the correct answers. If you got the all answers correctly, congratulations. Let us move on to another type of non-Mendelian inheritance pattern, the occurrence of multiple alleles. It should be obvious, based on its name, multiple alleles, that among the four postulates, it does not follow the occurrence of unit factors in pairs. We will delve deeper into that in a short while. Unlike in Mendelian inheritance, wherein a gene comes in the form of two alleles, in the case of multiple alleles, a single gene comes in the form of three alleles. A classic example showing the occurrence of multiple alleles is that of the human ABO blood groups. How many blood types are in existence? There are four. Type A, Type B, Type AB, and Type O. These four blood types are the phenotypes. How do we represent the corresponding genotypes for each phenotype? Do we write A, B, and O? The answer is no. Instead, we use I superscript A, I superscript B, and lowercase I, to designate the three alleles. But, what does I stand for? I stands for isoagglutinogen, another term for antigen. The allele IA is responsible for the production of A antigen. Allele IB is responsible for the production of B antigen. While allele I does not produce any detectable A or B antigens. Alleles IA and IB are both dominant alleles, while allele I is the recessive allele. Next, let us prick to another type of allelic interaction, called codominance. In codominance, the postulate on dominance over recessiveness is not followed. In codominance, heterozygotes exhibit a mixture of the phenotypic traits of both homozygotes. The definition might seem similar to that of incomplete dominance, but these two types of inheritance patterns are totally different. We will examine where the difference between the two lies as we go along our discussion. As in the case of multiple alleles, we will be using the human ABO blood group as an example. To illustrate the occurrence of codominance, let's say, for example, that a human male, who is of blood type B, married a woman with blood type A. What will be the resulting phenotypes? What blood types do we expect to see among the offsprings? The offspring with genotype IA and I will have blood type A. The offspring with genotype IB and I will have blood type B. The homozygous recessive offspring will have blood type O. But, the one with both dominant alleles IA and IB will have blood type AB. As you can see in the illustration, the red blood cell of an individual with blood type AB has both kinds of antigens, A and B, on its surface, in equal number. This table summarizes the genotypes and phenotypes formed by the different combinations of alleles. Now let's discuss the occurrence of dominant lethal allele. The presence of one copy of the dominant allele results in the death of an individual. Given this empty Punnett square, determine the resulting genotypes, and predict which of the offspring will eventually die. 3 out of 4 offsprings will die, and only one is normal and will live until old age, if no other misfortunes in life strike him or her. You might ask, do individuals who bear one copy of the dominant lethal allele die immediately, at birth, or a few days after birth? Individuals suffering from Huntington's disease, an example of inheritance pattern, which exhibits dominant lethal allele, live until the age of 40. Upon reaching 40, or even less, they die. Now, let us discuss sex-linked traits. Alleles that code for sex-linked traits are located on the sex chromosomes X and Y. Sex-linked traits coded by alleles on the X chromosome follow two inheritance patterns, dominant or recessive. We will be discussing the characteristics of X-linked dominant inheritance patterns first. Its distinguishing features are summarized in this table. Based on prevalence, there is equal likelihood that the inherited trait will affect males and females in a given generation. Most of the time, the affected mother will bear a son who has inherited that dominant X-linked trait. What is strange in this type of inheritance pattern is the transmission of the dominant trait from the unaffected father to all his daughters. And, since we are talking about a dominant trait, in which even one copy of the dominant allele is present, will result to the expression of that trait, there is no skipping of generation of that trait in a family pedigree. For example, in a family, the grandmother is affected by a dominant X-linked disease, her son will also be affected by that disease. When the son marries a woman who is normal and healthy, all his daughters will also have that disease. The trend will just continue as we go down the family tree. To further elaborate on the occurrence of X-linked dominant traits, refer to the following illustrations. Let's say, for example, that a certain disease, which is coded by the dominant allele A, is located on the X chromosome. A woman, who is affected by the disease, as shown in her genotype, married a man who is normal. Take a look at his genotype, the recessive form of gene A is present. Hence, the man does not have that disease, and is normal. The question is, what is the probability that they will have a son, who is affected by that disease? Or a daughter, who also has that disease? Take a look at the resulting genotypes, made from the union between an affected woman, and a normal man. There is 1 out of 2, or 50% chance, that a daughter will have that disease. Similarly, there is also 1 out of 2, or 50% chance, that a son will have that disease. Our example validates that the occurrence of the dominant trait is equal between males and females. Let us extend our example. If the affected son marries a normal woman, who is homozygous recessive for that trait, what will be the resulting genotypes? Which between the son or daughter will have that disease? As you can see, the father passed on the trait to all his daughters. The sons were unaffected, because the mother is also unaffected by the disease. If the affected daughter marries, and bears a child, later on, there is also a huge chance that her child will get that disease. Thus, proving that the trait does not skip generations. Let us proceed to identifying the features of X-linked recessive traits. As to frequency of occurrence, the trait could be found affecting more males than females. The recessive trait usually came from an unaffected mother, but when she bore a son, her son will be affected by the disease. What is peculiar in this type of inheritance pattern is that the trait is not passed on from a father to his son. Since the trait is recessive, and will require 2 copies of the recessive alleles before it could be expressed, skipping of generation along the family tree is expected. To further elaborate on the occurrence of X-linked recessive traits, take a look at the following illustrations. Remember that in order for a recessive trait to be expressed, it will require 2 copies of recessive alleles to be present. A woman, who is unaffected by the disease, but a carrier of that disease, was married to a normal male. When we say carrier, that individual is not affected by the disease, but carries along her X chromosome a copy of the recessive allele, responsible for the expression of the disease. Let us take a look at the genotypes of the offsprings. Both daughters are normal. But one of them is a carrier, due to the presence of a single copy, of the recessive allele. One son is normal, for its X chromosome carries the dominant allele. However, one son will be affected by the disease, even if only one copy of the recessive allele is present. Take note, that the allele is absent in Y chromosome, so there is no way, in which the dominant form of the allele could be present, and mask or stop the expression of the recessive allele. Hence, the recessive trait will be expressed, even if only a single copy of the recessive allele is present. Let us extend our example. When the affected son marries a normal woman, the resulting genotypes of the offsprings reveal, that none of them will have that disease, thus proving that the recessive trait may skip generations. Let us now proceed to the last type of sex-linked inheritance pattern. Perhaps, among the three sex-linked inheritance patterns, Y-linked is the easiest to understand. Since Y chromosome is only present among males, the occurrence of a Y-linked trait will only be found among males.